All right, get ready for a deep dive that'll feel like Biology 101. We're going to be exploring those amazing building blocks of life, proteins, carbs, and lipids.
It's funny, huh? You hear those terms all the time, but most people don't realize how intricate these molecules are. They're like the hidden machinery of life.
So true. Let's start with proteins. Now, I remember from way back when, proteins are made of these things called amino acids, right? Can you break that down for us?
Totally. Think of amino acids like the letters of the alphabet.
Okay, I'm following.
Just like you can arrange letters in different combos to make different words, you can link amino acids in specific sequences to make a huge variety of proteins.
So each protein is like a unique word spelled out with amino acids.
Exactly. And just like one wrong letter can totally change a word, even a single change in the amino acid sequence can totally change a protein's shape. And that changes how it works. It's all about precision.
Wow, it's like a secret molecular code. So what exactly are amino acids made of?
Well, each one has a central carbon atom bonded to four things: a hydrogen atom, an amide group, which is where the name comes from, a carboxyl group, and then a unique side chain they call the R group.
Our group, huh? Sounds important.
It is. It's what makes each of the 20 common amino acids different. Some are simple, some are more complex, some love water, some, totally hate it. And this variety in R groups is what makes proteins so versatile in how they're built and what they do.
Okay, starting to get the picture here. So how do these amino acids actually link up to, form proteins?
Through a special bond called a peptide bond. It's like a little handshake between two amino acids. When they join up, a water molecule is released and bam, peptide bond.
Oh, I like that. So two amino acids tie the knot, and water is the wedding favor.
Haha, yeah. And as more amino acids join in, they make a chain called a polypeptide. Polypeptides are like the rough drafts of proteins.
Got it. But if a polypeptide is just a chain of amino acids, how does it become, well, a functional protein?
Right.
With a specific shape. That's what determines what a protein actually does, right?
You're exactly right. And that's where things get even cooler. We need to talk about the four levels of protein structure: primary, secondary, tertiary, and quaternary.
Sounds kind of complicated.
It's actually pretty elegant. The primary structure is just the sequence of amino acids in the polypeptide chain, like beads on a string.
Okay, that's like the basic blueprint.
Exactly. Then secondary structure is where things start to twist. Hydrogen bonds between the amino acids make the polypeptide chain fold into repeating patterns.
Wait, those are the same bonds that hold water molecules together, right?
Precisely. They're weak on their own, but together they're strong. Like, think of a bunch of ants carrying a crumb way bigger than them. These hydrogen bonds in proteins make two common shapes: an alpha helix, which is like a coiled spring, and a beta pleated sheet, which looks kind of like a folded fan.
So we've got beads on a string, springs, and fans. What's next?
Tertiary structure. This folds those springs and fans even further into a unique 3D shape.
It's like origami, but on a molecular level.
Exactly. And this 3D shape is held together by a bunch of different interactions between the amino acid R groups, like those hydrophobic and hydrophilic things we talked about, and also ionic bonds and disulfide bridges, which are super strong.
Wow, it's wild how all those things work together.
It really is. And for proteins made of just one polypeptide chain, the tertiary structure is the final form. But some proteins are like complex machines with multiple parts. Each part is a separate polypeptide chain.
Ah, so that's where quaternary structure comes in.
You got it. Quaternary structure describes how those multiple chains come together to form a functional protein, like putting together different origami pieces to make a bigger sculpture. Hemoglobin, the protein that carries oxygen in our blood, is a classic example. It's got four separate chains, each with its own heme group to bind oxygen.
Wow, from beads on a string to like a complex oxygen carrying machine. It's mind-blowing. But before we go on, can I ask something?
Yes, sure, go ahead.
So we talked about how a protein's shape is crucial for it to do its job. What if something misses with that shape?
Great question. That's something called denaturation. Imagine you take that cool origami crane we talked about and you crumple it all up. That's kind of what denaturation does to a protein.
So it's like the protein loses its superpowers.
Exactly. Denaturation messes up the protein's structure so it unfolds and can't work right, like bending a key so it won't fit in a lock anymore.
Yikes. What can cause that?
All sorts of things. Heat is a big one. Think about cooking an egg. The heat denatures the proteins in the egg white, making them solid.
Ah, makes sense. So it's like the bonds holding the protein together can't take the heat.
Right. Changes in pH can do it too. Think about how lemon juice curdles milk. That's because the pH change denatures the milk proteins. And some chemicals can do it too, like detergents or solvents.
So these proteins are always trying to keep their shape in the middle of all these disruptors.
You got it. And this balance is what makes proteins so fascinating and vital to life, ready to move on to carbs, those energy
Yeah.
powerhouses.
Bring on the sugar rush.
Great. Just like proteins are chains of amino acids, carbs are built from chains of simple sugars called monosaccharides.
But glucose is one of those, right? Like the fuel our cells run on.
Which, you got it, glucose is the MVP of carbs. But there are others too, like fructose and galactose, each with its own sweetness and, personality.
Okay, so if monosaccharides are like individual sugar molecules, how do they make bigger carbs?
Through those special bonds again, but this time it's called a glycosidic bond. Just like peptide bonds link amino acids, glycosidic bonds link up monosaccharides.
So it's a different kind of handshake, but for sugars.
Yeah, and when two monosaccharides join, they make a disaccharide, like sucrose, which is just table sugar.
Oh, so that's what sucrose is.
Yep, it's a glucose and fructose team-up. But things get even more interesting when you link a bunch of monosaccharides together. That's when you get polysaccharides.
Polysaccharides, those are the big complex carbs, right? Like starch and cellulose.
Exactly. Starch, cellulose, and glycogen are all polysaccharides, and they're key for storing energy and giving structure.
Okay, let's unpack those. Let's start with starch. I know plants use it for energy, but what about its structure?
Starch is actually made of two types of glucose chains: amylose, which is a long coiled chain, and amylopectin, which is branched.
So amylose is like a tightly wound spool of thread, perfect for storage.
Exactly. It's the plant's pantry, full of energy for later. And amylopectin, with its branches, is like having a bunch of exits in a crowded place. It lets the plant release glucose quickly when it needs energy in a hurry.
Got it. Now, what about glycogen? It's like the animal version of starch, right?
Precisely. We store glycogen in our liver and muscles. It's even more branched than amylopectin, making it super efficient for quick energy.
It's like our emergency fuel reserve for when we gotta, like, run from danger.
Exactly. A super fast energy bar. Okay, and lastly, cellulose. It gives plants their structure, right? But what makes it different from starch and glycogen? Well, starch and glycogen are all about storing energy, but cellulose is all about strength. It's the main part of plant cell walls, giving them shape and letting plants stand tall.
It's like the scaffolding that holds the whole plant up.
Right, and the secret to its strength is its structure. Cellulose chains are long and straight, unlike the coiled or branched ones we talked about. And these straight chains bunch up to make super strong fibers held together by, you guessed it, hydrogen bonds.
Wow, those tiny bonds make the whole plant strong. That's amazing.
It really shows the power of teamwork at a molecular level. But we have one more group of biomolecules, lipids, ready to dive into the world of fats, oils, and all that.
Yeah, let's do it. I'm ready to learn why lipids are so important, even though they get a bad rap sometimes.
Awesome. Because even though we hear about the downsides of fat, lipids are crucial. They store energy long-term, provide insulation, and even form the base of our cell membranes.
Okay, color me intrigued. What exactly is a lipid?
Lipids are a diverse group, but they all have one thing in common. They don't like water, they're hydrophobic, like water-fearing. Think about how oil and water separate. That's because oil is a lipid.
Right. And triglycerides are a type of lipid, right?
Yeah.
Can you remind me about their structure?
Sure. Triglycerides are the champs of energy storage. They're made of a glycerol molecule, which is like the backbone, and then 3 fatty acid tails attached to it.
It's like a glycerol head with 3 long, wiggly tails.
Exactly. And those fatty acid tails are what make triglycerides hydrophobic. They have long chains of carbon and hydrogen atoms that don't mix well with water. That's why fats and oils clump together. They're avoiding water.
Makes sense. Now, what about phospholipids? They're a big deal in cell membranes, right?
Yeah, they're like the architects of cell membranes. They're similar to triglycerides, but one of those fatty acid tails is swapped out for a phosphate group. And that phosphate group actually loves water.
Whoa, so it's like a triglyceride with a split personality?
For sure.
One side loves water, the other hates it.
Exactly. That weird structure makes phospholipids perfect for building cell membranes. In a watery environment, like inside a cell, they line up in a double layer. Their water-loving phosphate heads face out, and their water-fearing tails hide inside.
So it's like a double-layered shield protecting the cell.
Yeah. Right. The phospholipid bilayer controls what goes in and out of the cell, like a gatekeeper.
Fascinating. It's amazing how one small change can have such a big effect.
For sure. And we can't forget cholesterol, another important lipid that sometimes gets a bad rap.
Tell me about cholesterol. What does it do in the membrane?
Cholesterol has a unique structure with a ring-shaped part and a hydrocarbon tail. It's flat and rigid, which lets it slide between the phospholipids in the membrane.
Oh, so it's like a tiny wedge adjusting the membrane's properties.
Exactly. Cholesterol helps keep the membrane from getting too stiff or too fluid so it can do its job right.
Like a membrane mechanic. It's wild how all these different lipids work together to keep our cells happy.
Yeah, it is. Just like with proteins and carbs, there are tests scientists use to detect and study these molecules. It's like molecular detective work, figuring out what's there and how much of it.
Ooh, that sounds cool. Can you give me some examples of those tests?
Of course. First up, the Benedict's test, which is used to detect reducing sugars like glucose. It involves adding a blue liquid to a sample and heating it. If there are reducing sugars, the color changes. Could be green, yellow, orange, or even brick red, depending on how much sugar is there.
So it's like a color-changing magic trick that reveals the sugar. Awesome. What about starch? How do they detect that?
For starch, we use the iodine test. Super easy. You just add iodine solution to the sample. If there's starch, it turns blue-black.
That one sounds pretty straightforward. What about proteins?
For proteins, there's the Biuret test. You add a couple of things to the sample. If there are proteins, it turns purple, showing those peptide bonds.
And lastly, how do they find those sneaky lipids?
The emulsion test. You mix the sample with ethanol, then add water. If there are lipids, a cloudy, milky emulsion will form at the top. Busted.
Wow, we got color-changing magic, blue-black solutions, purple hues, and milky emulsions. It's a whole chemistry show.
It is. These tests are super important tools for scientists who study the building blocks of life.
This has been amazing. We've covered so much, from the basic structures of proteins, carbs, and lipids, to the tests to detect them. But I feel like we're just getting started.
There's always more to learn, but I think we've got a solid foundation now. And I can't wait to dive deeper into those details in your source material. It's going to be good. It really is amazing, huh? How these seemingly simple molecules can be so complex.
Yeah, no kidding. So we've talked about the basics of proteins, carbs, and lipids. You know, their structures, what they do, and how to find them. But I want to go a little deeper now. My source material mentions something about alpha and beta-glycosidic bonds. What's the difference?
Good question. It all comes down to how the bond is oriented. Think of it like holding hands with someone. Your thumbs can either both be pointing up or one up and one down, right?
Okay, yeah.
Same handshake, but a slightly different position.
So it's a tiny difference in how those sugar molecules are linked.
Exactly. But those tiny differences can make a huge impact on how our bodies digest carbs. For example, starch has those alpha-glycosidic bonds, and our digestive system breaks it down easily. But cellulose, with its beta-glycosidic bonds, is way harder for us to digest.
So that's why we can get energy from potatoes, but not from eating grass, even though they're both made of glucose.
Exactly. We just don't have the right enzymes to break those beta bonds in cellulose. Some animals, like cows and termites, have gut bacteria that do the job for them, though.
That's so cool. It's crazy how just changing the bond a little bit can make such a big difference for digestion. I'm also curious about those different bonds that give proteins their shapes, like we talked about ionic bonds, disulfide bonds, and those hydrophobic and hydrophilic interactions. Can we break those down a bit more?
Of course. Remember, the tertiary structure of a protein, that cool 3D shape, is all thanks to different interactions between those amino acid R groups.
Right.
So ionic bonds first. Imagine you have two magnets, one positive, one negative. They just want to stick together, right?
Yeah.
That's kind of what happens in proteins. Some amino acid R groups have positive charges, some negative. And when they get close, bam, ionic bond. It helps stabilize the protein's shape.
Got it. Now, what about those disulfide bonds? They have something to do with cysteine, right?
Yes. Disulfide bonds are special because they're covalent bonds. That means they share electrons.
So they're stronger than ionic bonds.
Yeah, you got it. When two cysteines get close enough, their sulfur atoms can form a disulfide bond, like a little molecular staple holding the protein together.
Cool. Okay, now for hydrophobic and hydrophilic interactions. Those are always a little tricky for me.
I know, right? Think of it this way. Hydrophobic means afraid of water, and hydrophilic means loves water.
Right, like we talked about with lipids.
Exactly. Proteins have both kinds of regions, depending on their amino acids. So inside a cell, which is watery, the hydrophobic parts try to hide from the water. They bunch up on the inside of the protein.
They're trying to stay dry.
Yep. And the hydrophilic parts, they love the water. They stick to the outside of the protein. All this back and forth Helps the protein fold up into the right shape and stay stable.
It's like a little dance with
Uh,
different parts of the protein trying to get comfortable.
I like that. So all those interactions, ionic bonds, disulfide bonds, hydrophobic and hydrophilic things, and even those hydrogen bonds, they work together to make that unique protein shape and make sure it can do its job.
It's incredible, like a perfectly choreographed molecular ballet. It makes me think about those three polysaccharides, starch, glycogen, and cellulose. What are their specific roles again?
Let's see, starch first. The plant's energy stash, its structure is all about storing as much glucose as possible in a tiny space. Amylose, all coiled up, is really good at that.
And amylopectin, with the branches, lets the plant grab that glucose fast when it needs it, right?
Yep, more surface area for enzymes to break it down.
Makes sense.
Yeah.
Now, glycogen. It's also branched, even more than amylopectin. Why is that?
Animals need quick energy bursts, more than plants. Like for sprinting or fighting off danger, glycogen's structure makes it super fast at releasing glucose for when you need it now.
It's like having a fuel tank ready to go.
Exactly. And finally, cellulose.
Mm.
Not about energy at all, right? It's all about strength.
Yeah, it's what keeps plants stiff and upright.
Right. It forms these long chains that bundle up into super strong fibers. And guess what? It's those hydrogen bonds again, acting like tiny stitches to hold it all together.
So even though they're all made of glucose, those three polysaccharides do completely different things.
Amazing, right? Just those little differences in bonding make all the difference for their properties and what they do.
Totally. Okay, I want to shift gears a bit. We talked about how messing up a protein's shape can denature it and make it useless. How do scientists actually study that process? My notes mention something called biochemical tests.
Ah, yes. Biochemical tests are like a toolbox for studying these molecules. They let scientists find them, identify them, and measure them. And yeah, they can even be used to study denaturation.
Cool. Tell me more about how they use these tests to study denaturation.
Sure. Remember the Biuret test for proteins? Well, when a protein gets denatured, it unfolds, right? And that exposes those peptide bonds that the Biuret reagent loves to bind to. So if you do the test on a denatured protein, you'll often see a much brighter purple color. It's like the protein's shouting, "Hey, look at all my peptide bonds."
So it's like a visual clue that denaturation happened.
Exactly. And other tests, like looking at how light absorption or fluorescence changes, can also tell us if a protein is unfolding.
That's so cool. It's like these tests give us X-ray vision for the molecular world.
You got it. That's what's so amazing about science. We're always coming up with new ways to see what we couldn't see before.
Speaking of mysteries, there's one thing I'm still confused about. We talked about how cholesterol is important for cell membranes, but it can also be a problem, right? My source material mentions a link between cholesterol and heart problems.
Ah, good point. Cholesterol is essential, but too much of it is bad news. It's all about balance. When there's too much cholesterol in your blood, it can build up in your arteries, forming plaque.
So it's like clogging up the pipes.
Exactly. That plaque narrows the arteries so blood can't flow as well, and that increases the risk of heart attacks and strokes.
So it's kind of like a double-edged sword. We need it, but too much is bad.
Right. That's why it's so important to keep your cholesterol levels healthy, through diet, exercise, and maybe medication if you need it.
Wow. It's crazy how these tiny molecules can have such a big impact on our health.
It really is. It shows how important it is to understand not just what these biomolecules are, but how they interact with each other and with our bodies.
This has been an amazing journey. We've explored proteins, carbs, and lipids, from their structures to their functions, and even how they relate to our health.
And we've seen how scientists are using this knowledge to develop incredible new therapies and technologies.
It really is amazing how much these tiny things affect us. Okay, so we've talked about how messing with a protein's shape can, break it. But how do scientists actually study that? I read that they use something called biochemical tests.
Right. Biochemical tests are like a toolkit for studying molecules. You can use them to find molecules, figure out what they are, and even measure how much is there. And yeah, they can be used to study denaturation too.
Cool. So how do those tests help them study denaturation?
Well, remember the Biuret test, the one for proteins.
Mhm.
When a protein unfolds, like when it's denatured, it exposes those peptide bonds, and the Biuret reagent really likes to bind to those. So if you do the test on a denatured protein, the purple color is usually way more intense.
That's like the protein saying, look at all my peptide bonds, I'm unfolded.
Uh-huh, exactly. And there are other tests too, like you can look at how light absorption or fluorescence changes, and that can tell you if a protein's unfolding.
That's amazing. It's like we can actually see what's happening to the molecules.
It is pretty cool. And that's what's so exciting about science. We're always finding new ways to see things we couldn't see before.
I feel like we've learned so much on this deep dive. I'm definitely not going to look at a potato or a piece of celery the same way again.
Me neither. It's amazing to think about all the intricate processes happening at the molecular level all the time to keep us alive and functioning.
It's definitely given me a whole new appreciation for the complexity of life. Thanks for guiding us through this incredible molecular world.
My pleasure. It's been fun exploring these building blocks with you.
That's it for this deep dive. Hope you enjoyed it. Until next time, stay